What's up, everybody, penguins? So it is Friday, which means it's time for FRQ Fridays. So we're going to do the second FRQ from the 2021 exam. We did last week, we went ahead and we did the 2021 number 1. So this week we're going to do 2021 number 2. Okay. So without further ado, let me flip my camera so we can get going. So the geneticists investigated mode of inheritance of a rare disorder that affects the glucose metabolism and first shows symptoms in adulthood. The geneticists studied a family in which some individuals of generations 2 and 3 are known to have the disorder. And so you know they have the disorder because if you look at the pedigree, you can see that they're shaded in, which means that they're affected. So you can see that there's individuals in 2 and 3 generations that are known to have the disorder. Based on the pedigree and figure 1, the geneticists concluded that the disorder arose in individual 2 and that it was caused by mutation mitochondrial DNA. So just in case you're having trouble following everything, each of your generations are labeled over here, 1, 2, 3, and 4. And whenever they're identifying a person that's in it, you're going to identify it based on the row and then the number. So like this individual right here is going to be 2, 2. So you would write it out as 2, 2, because we're in the second generation, second individual. Okay, so that's kind of how you would name that. And so if you're looking at it, it also is giving you the key, but the females are going to be circles and the males are going to be squares. And again, this is male assigned at birth and female assigned at birth. They're also giving us 2 different data tables, right? So we have this thing that's looking at 8 different individuals. It also looks at their blood glucose level. And we're also given some information over here that tells us about whether they're normal, at risk, or whether they are affected, okay? So our first question that we have is the disorder alters glucose metabolism. Describe the atoms and types of bonds in a glucose molecule. So you have to think to yourself, okay, glucose. Glucose is a carbohydrate. What makes up a carbohydrate? So we have carbon, we have hydrogen, and we have oxygen. Like the name tells you what it is. Carbon, hydrate, oxygen, hydrogen. So we know that it has carbon, hydrogen, and oxygen in it. And then it also says we have to give it the type of bonds. So what kinds of bonds do we see in our macromolecules? We're mostly seeing covalent bonds. We're seeing those bonds in which something is sharing that valence electron, right? 2 different atoms share that valence electron. We know that it's polar if it's unequal sharing. We know that it's non-polar if it's equal sharing. So like carbon is non-polar and carbon-oxygen would be polar, okay? Now that went a little bit above what you probably had to write, but I was just kind of giving a little bit of information since we were already on the topic. So let's flip it over and see if we're right. So it says the atoms are carbon, hydrogen, oxygen, and they're held together by covalent bonds. Okay. And that right there was just a single point. You just had to be able to do that. Okay. And as I told y'all last time, part A is going to be your biology question. It's going to be asking you something about your biology knowledge. It's going to make sure that you understand the biology. Okay. As we move into part B and C and D, we're going to move into the experimental part of it. Okay. In which we're going to be doing a graph. Since we're looking at number 2, we're always going to have to graph on your second question. That graph can be a line graph. It could be a bar graph. It could be a box and whiskers, which we haven't seen before. It could be a scatter plot. It could be a histogram. It could be any number of graphs. And they just are asking you to graph one of them. And you have to be able to identify which kind of graph we're going to do. So using our template, in the space provided on your response, construct an appropriately labeled graph based on the data in table 1. So one thing we have to notice is these individuals, right? Is there anything that really ties them to each other that makes some type of linear scale? No, they're individuals, right? There's no really connection. I'm looking for a connection between them. So this is what we call categorical data. Because of the fact that this is categorical data, this tells me this is going to be a bar graph, okay? Looking here, I can see this plus or minus. That plus or minus is telling me my error bar. My mean is going to be 170 plus 15, and then 170 minus 15 is where I'm going to make those error bars, okay? And we're going to look at that in just a second when we make our graph, okay? So here's our graph, right? So the first thing we're going to do is label our axes. So we have individuals on our X, and we have our average blood glucose on our Y. Now notice, I have my units there, okay? The thing that I always tell students to do is whatever you have in your data table, you can put it directly into your graph. So individual, average blood glucose level, units, plus or minus, SEX, that gives you your other axes. Okay, so use exactly what they've given you and you will not forget anything because it's all been given to you in the chart already. Okay, so we go ahead and we graph our first individual, we graph our second individual, third, fourth, fifth, sixth, on and so forth. Okay, so now we have all of our individuals graphed, and don't get nitpicky with me. I did my best graphing on a computer. So are we done? Well, no, because we still have to make our error bars. Okay, so you're then going to put your error bar going above and below that amount. So as we said before, 170 plus 15, 170 minus 15 based on my data, right? So it said that individual 1 should have been 170 plus or minus 15, okay? So we're going to just graph that, okay? That in itself is worth 3 points, okay? Your axes are usually a point. The scaling of it being appropriate, labeling them correctly, having units, all that's usually a single point. Your second point is usually, did you correctly graph the information? Okay, so if you haven't correctly graphed the information, you're probably not going to get that point. If you make the wrong graph, you're probably not going to get that point. Okay, and then the third point would be our error bars. Okay, so usually graphs are worth 3. If you're running short on time, go ahead and just label your axes, and that guarantees you that, at least that 1 point. Okay, and then if you don't have enough time to completely do everything, just go ahead and just label your axes. Okay, you will be writing in pen on the exam. But what you're allowed to do is you could probably write in pencil to get your general lines of where you're going to be putting everything. You know, oftentimes we make errors when we're trying to scale an axis. You can use a pencil, scale your axes, and then overwrite it with pen if you have time. If you don't have time, do not worry about changing that pencil into pen. We as readers will still grade it. Because of the fact that things are being scanned in this year, I'm not 100% positive how the pencil's going to show up on the scan. Okay, so if you can replace the pencil with pen, I highly recommend doing that, but you are able to technically use that pencil. There's a question that popped in, but the problem is I have so many questions that are in my stories, I can't really figure out which one it is. So here is the official, what was on the scoring guidelines. They had this graph, which was made by a certain student. And then, of course, we've got our point distribution, axis labels, plotting bar graph, modified bar graph, and error bars. If you're wondering what a modified bar graph looks like, a modified bar graph looks like you've put a data point at each of the means, and then you have the error bars coming above and below that. Okay. Now, I see there's a bunch of questions coming into the chat. We're doing FRQ Friday. We're going over this FRQ, and I can do questions afterward, but right now I'm focusing on the FRQ. So they then tell us that we're supposed to determine one individual who is at risk of developing the disorder and has a significantly different blood glucose level than that of individual 1. Okay, so number 1, we have to think through here and say, okay, well, who is at risk? Anyone that is 140 to 199 is going to be at risk. So we draw a little, I drew these little bars. You wouldn't really do that, but I'm doing this just so I can visualize it for you. So between 140 and 199, there's 4 different individuals. Okay. I've got individual 1, 2, 3, and 4. And now I need to figure out, well, okay, these are people that are at risk. Who is also statistically significantly different than 1, right? So I'm going to look at where my error bar is for 1. Okay, my error bar from 1 overlaps with individual 2 and it overlaps with individual 4. Okay, that tells me that these 2 data points are not statistically significant. Okay, you want the ones that do not overlap. So that would be individual 1 and 3 do not have that overlap. And so we know that individual 3 is significantly different. And then also is going to be at risk for developing that disorder. Okay, so now part C. Based on our pedigree, which they gave us this pedigree at the beginning, identify all individuals in generation 4 who can pass on the mutation to their children. Okay. Now, remember, the prompt told me that the trait was mitochondrial inheritance. Okay. That they believed that it was going to be due to mitochondrial inheritance. The mitochondria is going to be in the egg. So the egg producing organism is the one that's going to be able to pass this on. Okay, so based on our pedigree, we're going to look at the females. The females are the ones that are able to pass on this trait. So a female would pass it on to all of their offspring. If you have an affected male, that's not going to be able to pass it on to their offspring because they don't donate the mitochondria. Okay, so that's what we're looking at here, is that this should be mitochondrial inheritance, right? So prompt stated is mitochondrial inheritance. So we then are looking at identify individuals in generation 4. So we're looking at here, these individuals down here, who is going to be able to pass it on. Okay, so here we have 3-2 has the trait and is going to pass on that trait to 1, right? Now, it got it, true. Can this individual pass it on? Well, yes, because that is a female. Okay, so if you look here, number 4 doesn't have any offspring, so we don't really worry about that one. 6, again, we have an affected female, which means that number 2 is affected, but it's also a female, so it can pass it on. Looking again, this male is going to be affected because it has an affected mother, but it is a male, so the males cannot pass on that trait, so we're not going to circle 3. Again, affected female, affected female child is going to be affected. So we're going to say that individuals 4-1, 4-2, and 4-4 are all able to pass on that mutation to their children. Okay. So what you're seeing here is you're having to apply some type of knowledge to extract information, right? You're going to have to know what mitochondrial inheritance is and then be able to conceptualize and work through it in your mind to be able to answer the question they're asking. So the last thing we're going to have to do is we now need to defend something, right? There's some type of prediction, or we're going to have to justify someone's prediction, make a hypothesis, all that fun stuff. So based on the fact that individual 2 is affected, a student claims that the disorder is inherited in an X-linked recessive pattern. Based on the student's claim, so we're keeping in mind what the student's claim is, and I need to say, okay, if this student is right, I need to say what are going to be the individuals in 3 that will be affected by the disorder, right? So looking here, affected, right? We need to figure out who's going to be affected in 3. Well, if this is sex linked, right, and it's got on the X chromosome, you need to have 2 X chromosomes in order to have an affected female, right? This male is not affected. The one and only X that this male had did not have the recessive trait on it. So there's no way the individual 3-2 could be affected because of the fact that there's no way for this individual to get 2 of those faulty X chromosomes. So if we look here at 3-4, could that female have passed on one of her X's to this male? Yes, it's completely plausible. Again, we're looking at a female that's affected. There's no way that you could have an affected female because there's not an affected father. And then if we look here, we have an affected male, and that's plausible, okay? So individuals 2 and 8, 3-2, 3-8, are going to be the ones that are going to be able to be affected by the disorder if it was X-linked recessive. And our reasoning is that the data does not support the claim because of the fact that individuals 3-2 and 3-6 have the disorder, and they should not be able to have the disorder because of the fact that the father does not have the disorder. And then also you could talk about that the data doesn't support the mitochondrial inheritance because individuals, like the sons, are not the only ones that are affected. So that is FRQ number 2. Next Friday, we'll go live again to do numbers 3 and 4. Okay. I want to remind you that on Sunday at 4:00 p.m. Eastern Time, I'll be going live with Marco Learning on their YouTube channel. We're going to go over Unit 7. And then next weekend on Sunday, again, 4 p.m. Eastern Time, we'll go live with Marco Learning to do Unit 8 review. Okay, so the questions that came through the chat, I'm going to go ahead and answer those. I don't know if the person's still in here. I hope they are, but if not, so someone asked about null hypothesis. A null hypothesis is stating that the experimental treatment had no effect. So whatever it was we used is not going to have an effect on the outcome. So let's think about like caffeine and bees, right? So we would say that caffeine has no effect on the memory of bees because we would find that the control group and the group that had caffeine would have the exact same mean data, or their error bars would overlap. That's saying our null hypothesis. Explain the difference between dependent and independent variables while drawing our graph. So when you're thinking about dependent variable versus independent variable, dependent variable is the thing that you are changing, right? So in our experiment, we looked at 8 different individuals. So we changed which individual we were looking at. If we're thinking about caffeine and the bees, that would be the time. We did it at 10 minutes and we did it at 24 hours. And if you're wondering what question I'm referring to, there's a bee caffeine question that's like 2016, number 1, I think, in which that came up. Versus the, did I say that wrong? I said that wrong. I'm sorry, y'all. Independent variable is what we were changing, and the dependent variable is what changed because of that. I'm sorry, y'all. I'm tired and running around in circles as always. So let me say that again since I said it wrong originally. independent variable is what's going to be on your x-axis, and it's what we changed. So the individual numbers were the independent variable. And then what changed because of that was the concentration that they had. So that would be your dependent variable. Sorry, y'all. So I don't think there's any other questions. There's someone that popped something into the question box, but there's no way for me to see what's in the question box because of the fact that I've got so many other things that are on my story question box, and they all coagulate together. So I hope y'all have a wonderful weekend. If you've had payments or just success, bye y'all.